So, in this lecture module, I shall give the overview of mammalian cell to the non-biologist, and at the same time I shall give that utility of microfluidics platform to unravel that cellular behavior like in vivo situation, and at the same time cell at the single cell level how it behaves, and to unravel their physiological and other chemical and biochemical cues. Now, if we look for that cells, these are the unit of all living organisms, is comprises of that different types of molecules, and so this is the cell. This is the unit of living organisms, and it obeys that all the physical and biophysicochemical laws of nature, which is obeyed by that non-living matters also. So, these cells are organized to tissues, then tissues are organized to organ, then organ to organ system, then whole organisms. This hierarchy of organisms obeyed in all types of higher organisms, and if you look for the cells, is composed of molecules and atoms. If we consider the dimension of the cells, like say animal cell, plant cell is in the range of around say 10 micrometer to 100 micrometer, whereas in the bacterial cells is the range of 1 to 2 micrometer, and some cell organelles is in the range of 1 to 2 micrometer. These cells are organized to whole animal, which is the dimensions are around that 1 meter, and that molecules is in the range of say 0.1 nanometer to say 4 nanometer. Now, why these cells are have a diameter of around 10 to 100 micrometer, whereas that mammals, the whole organisms, the dimension is very big 1 meter. Due to that unicellular system, they can take that material by diffusion through high surface area, whereas in the animal system or whole organisms, their surface area to volume ratio decreases, that is aspect ratio decreases, and they cannot get that materials by diffusion only. So, for this higher organism develops that sophisticated transport system to transport that materials actively, which is compensated by that diffusion limitation for passive transport. Now, if we look for that biochemical reactions which are manifested to that cellular behavior, what is the time scale? So, if we look for that your enzymatic reactions, like say it occurs at that millisecond level, where the signal transductions occurs at that nanosecond level. What is the signal transduction? Signal transduction means it may be a chemical entity or may be biophysical entity, which transmit the signal from surface, cell surface or cell membrane to the interior of the cytoplasm, then goes to the nucleus. Then it is this message is processed, then it is manifested in the terms of proteins. So, this signal transduction takes around say nanosecond level, whereas that protein expression and that ultimately enzyme activity the millisecond levels, and all these events goes to manifestation of say some cell division. It in the bacterial system it took around 20 minutes, whereas in the eukaryotes like say HeLa cells or you say mammalian cells it takes around 20 minutes. So, you can see that how much scaling law occurs in the time scale, at the same time the dimension scale or the aspect ratio is mattering the things. Now, what are the molecules constitute a cell? Say 70% of the cell mass, basically it is water, then the remaining 28% are like that, it is comprised of the 4 macromolecules like say protein, nucleic acid, fats and carbohydrate, and the remaining 1 to 2% all the minerals basically. So, if we look for that dry weight of cell, say around 50% of the dry weight is protein, whereas DNA comprises only 3%, RNA is around 20%, lipids are around 10%, and the remaining percentage are comprised of your carbohydrate, minerals, etc. So, if you look for that protein and DNA, these are the one at the higher end and other in the lower end. They are the main contributing molecules in a living cell. They controls whole the organism's activity. Now, you look for the protein, which is the major part of the dry weight constituents. So, protein is made up of amino acids. So, what is amino acid? It is a deuterionic compounds. It has a NH2 group and it is a carboxylic group, and this is the side chain R groups. So, depending upon the nature of the R groups, these amino acids are grouped, and we have 20 proteogenic amino acids. These R groups are hydrophobic, polar, non-polar, anionic and cationic nature. These amino acids are linked by that peptide bond, like say another amino acid, that peptide bond is formed, and due to the restriction of the peptide bond, that in that is carbons are these peptide chains have that conformation in alpha helix and beta pleated sheet structure. So, this is that is that primary sequence of the amino acids, how these amino acids are linked by the peptide bond, and then this is go to the secondary structures. Now, secondary structure elements like alpha helix and beta pleated sheet, they are arranged 3 dimensionally to make the tertiary structures, and main contributing force for the tertiary structures are your hydrophilic, ionic, hydrophobic and weak van der Waals forces and hydrogen bonds. And this tertiary structure is the real active form of the structure, which imparts that activities of the proteins. The tertiary structure of proteins again goes to quaternary structure, or you can tell the supramolecular structure by that aggregation. This quaternary structure, if you can look for that, say example is myoglobin, which carries that oxygen molecule, it is a subunit, 1 subunit structures. Within 4 subunit structure like say alpha 2 and beta 2, the quaternary structures which carries the oxygen that is present in the RBCs. And these proteins are main workhorse of the cell. So, like say it has these molecules has a structural activity, means like say filamentous proteins are mainly structures, where the globular proteins they have a lot of functions, like say starting from that receptor function, transport function, enzymatic function. Then these proteins acts as a molecular machines basically, and their efficiency of the machines from 50% to 100%. They convert that mechanical energy to chemical energy and chemical energy to mechanical energy. So, here we can see that different proteins of different sizes. This is say insulin, glucagon, these are acts as a hormone. This is cytochrome C, acts as the electron transport carrier. Calmodulin acts as calcium binding proteins. In that way, troponin acts in the actin-myosin, that is your stretch activity. In that way, that is hemoglobin is your oxygen carrier protein. This is aspartate carbon transferase, and glutamine synthesis are the enzymes, and this is immunoglobulin, that is it acts that antigen binding which protects our body from that external threats like say bacterial or microbial threats. So, what is nucleic acid, which comprises around 20% RNA and 30% DNA. This nucleic acid basic building block is nucleotide. What is nucleotide? This is a comprises of base. These bases are purine and pyrimidine. These purines are adenine, guanine, and pyrimidines are thymine and cytosine. So, this base is linked to pentose sugar, that is ribose, and one phosphate is linked to the 5th position of that ribose sugar. So, this is called the nucleotide. Now, these nucleotides are linked by the phosphodiester bond, like say so, in that way all the bases are linked by the phosphodiester bond. This is 5 prime end and this is your 3 prime end. In DNA, these strands are antiparallel, and this phosphodiester backbone is the backbone of DNA. Is the backbone of DNA is the phosphodiester bond, and that ATGC they are linked by that A is complementary to T by 2 hydrogen bonds, G is complementary to C, they are linked by that 3 hydrogen bonds. So, this ATGC pairs these are the means nucleotides, and these 3 nucleotide means sequence makes the genetic codon. So, these genetic codons are the carrier, means it is the message is stored in the form of genetic codes. And this, what the difference between DNA and RNA? RNA is that they have a hydroxyl group 2 positions, and these hydroxyl groups makes that RNA is unstable, whereas DNA has a no hydroxyl groups in the position. This is the deoxyribose sugar. So, that is a stable one, and this DNA is a double helix, the antiparallel double helix, and this carries that genetic information in the form of that genetic code. And in human system around say 98% of the genetic codes are non-coding for the proteins. This is the central dogma of our living system. So, how that the linear code of DNA is transcribed to messenger RNA, then this is translated to proteins. So, now if you look for the locations, this translation is going on in that is your nucleus messenger RNA, and it is transported to cytoplasm with the help of ribosome, tRNA and amino acids translated to protein, which is the main workhorse of that cell. Now, if we look for that next molecule, that is carbohydrate, which is the main energy source of the cell. Say these are the varieties of carbohydrates are there, monosaccharide, disaccharide and oligosaccharides and polysaccharides. And in disaccharides, trisaccharides, they are linked by that glycosidic bonds, and they are in the oligosaccharides, there may be from 3 to 10 by glycosidic bonds, and when their members are growing larger, we are telling these are the complex polysaccharides. And these carbohydrates are highly branched. In animal system, they are stored as a glycogen, whereas in the plant system, they store as a starch, cellulose, hemicellulose, etc. Next molecule is a lipid, which comprises 10% of the dry weight. So, these are the amphipathic molecules, which have a hydrophilic head groups and hydrophobic tail. This is one, and this is your, this is triglycerides, this is phosphoglycerides, these are the fatty acids, and these triglycerides acts as a oil what we are taking in our daily life, and phosphoglycerides are the main components of membrane. And what is membrane? It is that boundary of the cells from the external environment to the internal environment. So, these are the macromolecules. Now, how these macromolecules are arranged to make that organelles or of the living system. So, these are arranged by self assembly. So, what is self assembly? So, it is a non-template driven process. It is a non-template driven process, but the information present in the molecule itself, when the molecules comes together by diffusion, like an example of lock and key model, they binds each other by hydrophobic interaction by losing the water molecules tightly, and in that way that they mix that macromolecular structure to supramolecular structures. So, like in this example, we can show that nucleic acids binds with protein histone, they makes that chromatin, which is in the chromosome, that is present in the nucleus. That globular proteins like say actins, they make that actin filament, which is the cytoskeleton protein. Then tubulins are formed from the monomers, they make that large macromolecular structures for the which are used for transport system, intracellular system, as well as is the it helps the stability of that membrane system. Then lipids and proteins is the two different class of molecules. They are aggregated or organized or self assembled, we can tell to make that membrane. So, this is self assembly, it is one of the manifestation of the living system. So, what are the attributes or properties of the cells by which we can tell that it is a living cell? So, first attribute is your production of two identical replicas of DNA from one original DNA molecule to sustain that biological inheritance, means from one cell to its progeny, that whole the information is percolated by that your transcription of the DNA without much error. This is self replication. Then we are coming growth and metabolism. This is encompasses by that huge amount of biochemical and biophysical networks for the survival, growth, differentiation and ultimate apoptosis, that is your programmed cell death. And these networks are controlled by that feedback mechanism, but all the means complex manifestation of these cells cannot be explained simple by your feedback mechanism. So, next that manifestation of that cell is the transport system, both intracellular and that total cellular transport. In intracellular space, molecules are transported by that your passive diffusion process, but when the molecules are bigger than 30 kilodalton or say some vesicles, they cannot be transported by simple diffusion, it will take lot of time. So, that is why cells has developed their machinery to transport these macromolecules and vesicles by active transporter using that protein rods and that molecular machines like say actin, myosin, kinesins. Then we are coming that robustness of the cell. What is robustness? Say it is a robustness nature is that if we part up that internally or externally both physically and physicochemical ways, the cell has a property to sustain that steady state. So, we can take that or we can take that perturbations or you can take that robustness like homeostasis. Homeostasis means cell has a behavior to maintain that steady state if we change that temperature, pH, even if salt concentration within a tolerable limit. It can be a steady state or it can be a dynamic state. Dynamic state means like say with time independent way cell can synthesize or express some group of proteins. So, this is called circadian rhythms. So, these circadian rhythms will not be affected by that external or internal physical and physicochemical perturbation. So, this is that how this is maintained? This is maintained by high level of control system. So, these are the 4 manifestations we can tell that all the living cells has. Now, if you look for that what are the parts of a living system. So, this is that your unit of living system that is cell, here mainly describe that mammalian cells. This is the boundary between external and that internal of the cells is the boundary made up of your lipids, mainly phospholipids. Its composition is mainly 40-60 protein and lipids, and it harbors that lot of proteins which acts as a receptor, is a transporter and ion channels by which it can exchange the material from outside to inside. And this membrane is a hydrophobic in nature. So, that polar ionic molecules cannot transport easily. And this next is the cytoplasm, that is that whole that viscous like liquid like honey, and here that protein concentration is around 180 to say 400 milligram per ml, and this high concentration of protein gives that crowding nature of that cytoplasm and viscosity like honey, and molecules cannot transport from one place to another by simple diffusion. So, this is a diffusion restricted. So, for that how that big molecules will transport, as I told earlier also, these are the cytoskeleton elements like say tubulins by which that big molecules are transported by using their molecular machines. Now, we are coming to the nucleus, which is the head office of that cell, which contains that DNA, and that DNA length is around 2 meter. It is encompassed in the space of 4 micrometer, and they are highly packed basically. In the nucleus also there are a lot of proteins are there like say DNA polymerase, RNA polymerase and other transporters also, by which that nucleus can exchange that information from inside to outside. These are the organelles of endoplasmic reticulum, is a continuation of the outer membrane of nucleus, and here that proteins are translated with the help of ribosome. Most of the proteins are translated here and processed. These proteins are finally processed in that Golgi, which is the packaging system of the cells, and from that place that processed proteins and lipids and other complexes are transported various location of the cells very precisely. This is the mitochondria, that powerhouse of cell, where that metabolites are burned to carbon dioxide, and that heat generated for that burning is converted to energy called energy currency of the cells, ATP, and that ATP generated by using that is your molecular machine F0 F1 ATPase, its efficiency around 100%, which cannot be achievable by your man-made machine. And this is the lysosome, that is the recycle bin of a cell, where that used proteins which should be recycled here by that lot of lytic enzymes using that protease, lipase, your glycosidases, which are cleaved and they are recycled to make that other organisms, other organelles. In that way, all the organelles are arranged in the cytoplasm. So, we can tell that cytoplasm is the place where that high concentration of proteins and that it contains that cytoskeleton elements and your organelles. So, we can tell that this is a highly crowded space. We can take an example of that, say vehicles are transported in a highly traffic zone, where that pedestrians and like bicycle they can go easily, whereas big molecules cannot transport so easily. So, there needs that some transport system for directional transport or active transport through that is protein road tubulin by using your actin, myosin or kinesin or dynein type of your molecular machine to transport that cargoes from that one place to other. So, with this context we can tell that cell acts as a smart and a robust machine, because all the attributes of machine characteristics present in the cell, that is high level of automation, high level of integration of that molecular networks. On the other hand, we can compare the cell as a smart factory to give an understanding that how that cellular organelles are comparable with that organization of a highly automated modern day factory. So, this is that membrane which can be comparable to that your factory boundary. These are the gates of that boundary walls, means it is that transporters or ion channels, and this is that head office, means which transmit the message in the form of messenger RNA, or you can tell that this paper to that your cytosol, means other places. This is the endoplasmic reticulum, where these message is translated to that proteins and which are transported to the Golgi bodies processed form, and they are going to different places using that vehicles. This is the tubulins, which are the roads basically by which vehicles are goes and going to the different destinations. And this is that mitochondria, where that energy is generated by burning that fuels, that is going to carbon dioxide and ATP and energy. So, this analogy will give that idea of a cell as a factory, and earlier what you told, it is a machine property. In the omics area, we know that proteomics, genomics, metabolomics, transcriptomics, and nowadays it is coming the interactomes. With these omics and omes, we are able to get the knowledge of that how the cell works, what are the networks are involved, but there are a lot of things to tell in that biology. Like these are the some open ended questions in biology, like say inter and intramolecular interactions of biomolecular assembly leading to discrete as well as global function of the cells. Means say how that protein protein interaction, protein DNA interaction, lipid protein interaction, lipid carbohydrate interactions leads to that global functions of the cell, like say cell division, motility, differentiation and apoptosis. And how that integration of mechanical and chemical signals for various cellular functions, because we know that biochemical functions more than mechanical functions or the mechanical signal transduction. How these two processes are integrated for the cellular functions, and the discovery of new networks, or you can tell the hidden networks at the different level of organization, means what I want to mean that from an outsider we see that machine is working, and we do not know what are the hidden networks in the machine, and this is known by that who have prepared the machine, all the networks they know. But in a perspective of cell, we are the outsider, we do not know, because we have not created the cell, what are the hidden networks present at the different level of organization. So, these are yet to discover to know that complexity of the living system. Now, how microfluidic system will address these problems or related problems? First is that high level resolution and control of single and collective cell behavior. This can be addressed by that hydrodynamic trapping as well as microfluidic different types of valves to trap single cells or a group of cells together and study their behavior precisely by using high resolution microscopy. Then implication of micro confinement effect on cells. Here we want to define that if you put the cell in a different types of 3-dimensional biophysical cues which will deform that membrane as well as nucleus and manifested their phenotypic characteristics. Like say, if you put the cell in a microtube where that both nucleus and cellular membrane is deformed, how these things affect that cell viability, cell division, cell migration and apoptosis. Third point is that understanding that mechanosignals transduction and related biochemical networks for cellular functions, means how that we can generate that mechanical signal by using different types of fluid flow, and how these fluid flows generated mechanical signal transmitted or transformed to biochemical signal and affecting the cellular behavior. Like say in cancer situation, say cell are disseminated from that tumor to distant place through metastasis. Now, what that cellular means tumor stress involves to come out of the cells come out of the tumors. By using this platform we can unravel, and last of all that unraveling the cellular behavior by mimicking in vivo mechanical cues in cell tissue organ level. You take an example like say capillary bed network for endothelial system. We can mimic this in vivo system in vitro, and we can study that how that capillary bed networks, that fluid flow like say bloods can affect that endothelial cells and the related immune cells like T cell, B cells and other floating cells. Take another example in lymphatic node system, how that your lymphatic conduits where that antigen presenting cell and that T cell, B cells interact to recognize the particular T cells. This also can be mimicked in vitro system, other system like bone canaliculi and immunological and neurological synapses. All these things which are occurring in the living system at that capillary level or microscale level, we can mimic these systems in vitro to understand that biological phenomenon at the single cell level to your cell or multiple cell level by using this fluidic platform. Okay, next two classes we shall discuss all these phenomena sequentially to unravel that power of microfluidics platform for that biological questions. Okay.